Cloning by Kayla and Jaina.
So, to begin, what are clones and what is cloning? Well, clones are organisms that are exact genetic copies of each other, and cloning is the process of achieving this. The topic of cloning can be broken down into its processes and types, the impacts this scientific process has on society, its controversy, as well as its current accomplishments.
Cloning a gene. Cloning a gene involves scientists isolating and making exact copies of just one of an organism's genes. They do this by isolating DNA from an organism, then purifying the DNA and fragmenting it using a restriction enzyme. The restriction enzyme produces cuts and specific sequences to the DNA strand, isolating the desired section of DNA and creating cohesive ends. Later on, these cohesive ends allows for the DNA fragment to integrate into a plasmid, which is a small DNA molecule within a cell that is physically separated from the rest of the DNA and is able to replicate on its own.
A DNA fragment is able to integrate into a plasmid as the plasmid has been cleaved by the same restriction enzyme which creates the same cohesive ends as on the DNA fragment, allowing them to join together. The cohesive ends of the plasmid and the DNA fragment line up, and the enzyme called DNA ligase is used to create phosphodiester bonds between the nucleotides.
After this, the plasmid, with the DNA fragment incorporated, is added into a bacterial host cell, where it is then able to reproduce and replicate on its own when placed in a growth medium, allowing it to culture and reproduce.
The colony of cells that then contain the desired clone gene can be isolated and used for further study or to aid in medicinal purposes.
Cloning organisms. While gene cloning is the process of cloning only a single gene, cloning as most people think of it involves cloning all the genes in an entire organism. There are two different ways that this can be done.
A natural process in which this is done is through the formation of identical twins. In the first few days after the zygote is created, each half of the embryo continues dividing on its own, ultimately developing into two completely separate individuals. Since the two develop from the same fertilized egg, the resulting individuals are genetically identical.
The first way of artificial cloning is through artificial embryo twining, and this is a technique that mimics the natural process that occurs to produce identical twins. However, it is carried out in a petri dish. This method involves separating the early embryo into two individual cells, which are allowed to divide and develop in vitro. The embryos are then placed into a surrogate mother, where they are then able to finish developing, and the resulting organisms are then genetically identical.
Another method of cloning organisms is through somatic cell nuclear transfer, also known as SCNT. Broadly speaking, this means that a nucleus is removed from a healthy donor egg cell. The egg then becomes the host for a new nucleus from a somatic cell. The embryo that forms can be used to produce embryonic stem cells that are a genetic match to the donor of the nucleus, or can be implanted into a surrogate mother to create a cloned and genetically identical organism. However, in more detail, this process can be broken up into the steps defined by the acronym SCNT. The S and C stand for somatic cells, which is any body cell other than a reproductive cell. Every somatic cell has two complete sets of chromosomes, and the nucleus from one of these cells is taken and implanted into a reproductive egg cell.
N stands for nuclear, which refers to the nucleus of the cell, the organelle in the cell that contains the cell's DNA and is in charge of the plans for all cell functions. With the implanted nucleus, the daughter cells that the egg will produce will have the same genetic makeup as the implanted somatic cell's nucleus.
T is for transfer, which simply put is moving an object from one place to another, and in this case describes the movement of a nucleus from one cell into another.
Once a nucleus from a somatic cell is transplanted into an egg cell, it is stimulated with an electric current shock to induce cell division. After many divisions, this single cell forms a blastocyst, which is an early embryo made of around 100 cells, and this contains identical DNA to the original organism.
Natural clones versus artificial clones. In embryo cells all have two complete sets of chromosomes. The difference between fertilization for identical twins and SCNT lies in the source of those two sets. In natural fertilization, the sperm and egg each have one set of chromosomes. When they join, they grow into an embryo with two sets, one from the father and one from the mother.
In SCNT, the egg cell's single set of chromosomes is removed when the nucleus is taken out. It is replaced by the nucleus from a somatic cell, which already contains two complete sets of chromosomes. So in the resulting embryo, both sets of chromosomes come from the somatic cell.
Benefits of cloning genes. A benefit of cloning single genes is to aid in the production of recombinant proteins. Recombinant proteins are proteins encoded by recombinant DNA, which are DNA sequences that result from the use of lab techniques to bring together genetic material from more than one source, creating sequences that wouldn't otherwise be found in organisms.
Proteins that are normally produced in very small amounts, such as insulin in diabetics or blood clotting factor 7 in hemophiliacs, can then be made through these cloning techniques to create an outside source of the needed substance for different patients. Before gene cloning and protein production via recombinant DNA techniques, these molecules such as insulin were purified from animal or human blood. However, there were many problems with getting blood from these sources, such as slight differences in non-human proteins, as well as there was always the risk of diseases. Gene cloning of healthy genes provided an alternate option to be able to create these needed solutions.
Benefits of cloning organisms. These are some of the prominent benefits to cloning individual genes, but there are also many benefits to cloning entire organisms.
Cloning organisms is a key component to medicine that has helped to advance our knowledge of both diseases and healing.
For example, much of our knowledge of disease comes from studying animal models, who are genetically modified to carry certain diseases or gene mutations to portray a particular trait. Creating an organism with a specific disease or trait to then be able to study is much work, therefore cloning is used to help with this process.
Creating clones to use for medical research can help to reduce the amount of time needed to make a transgenic animal model, and the result creates a population of genetically identical animals to be studied. This process has increased the efficiency in medical research and has allowed us to make many scientific advances.
Controversy of cloning. Within cloning, there has been much controversy surrounding the cloning of embryos, specifically those of humans. This comes from the fact that though there have been successful clonings of entire organisms, they are very difficult to accomplish and often pose threat to the fetus and its survival. This is harmful since the fetuses of embryos cloned through SCNT rarely survive gestation and usually are born with birth defects from the complex procedure.
This brings rise to the controversy of modifying natural human processes, often harming the embryo and the surrogate. As seen through the 277 failed tries to create a cloned sheep, as well as attempts to produce a cloned monkey involving 100 embryos and 50 female monkeys, all of which did not result in a viable pregnancy. These safety factors to both the embryo and the surrogate provide arguments against the further continuation of research with cloning human embryos.
There have been numerous successes with cloning entire organisms. Perhaps the most famous of these was the cloning of Dolly the sheep in 1996, who was the first successfully cloned animal. Dolly was created through the process of SCNT, and after 6 days of normal growth as an embryo, she is transferred into a surrogate mother.
The pregnancy went without problems, and Dolly was born a lively lamb that did not suffer from any neonatal problems at birth, unlike many other cloned organisms. Dolly lived for 7 years until she died of sheep pulmonary adenomatosis, an incurable disease.
Another prominent success with cloning an organism was in 2002 with a cat called Cece, standing for carbon copy, or copycat. Cece was cloned by transplanting DNA through the SCNT process from Rainbow, a female 3-colored cat, into an egg cell whose nucleus had been removed, and then implanting this embryo into a surrogate mother.
She was not, however, physically identical to her DNA donor. The pattern on cat's coat is only partially genetically determined. It also depends on other factors during development, which caused Cece to not look entirely like her donor mother Rainbow. Out of 87 implanted donor embryos, Cece is the only one to survive, which is a higher success rate than that of creating Dolly the sheep, which took 277 tries.
Overall, cloning is an important scientific process that still has lots of room for exciting scientific growth in the future, but has also had many successes and scientific uses in the past. Though cloning is not in reality the way it is in the movies, it is a very neat and exciting molecular process.